Anyway, we'll start the class. So, welcome students for this foundation class. As you know, we have been discussing in the foundation class mathematics quite some time. We are doing that. Now, today we have one faculty by name Anisha, and she will be actually handling science, particularly biology. To start with, she will begin with biology. But today our file was not able to, she's not able to present her file due to some technical reason. But orally she can do the class. And Anisha, now you can go ahead.
Sure, sir. Okay, so today we are going to see about a very interesting lesson. I'm going to give you an overview. Sir will send the file and the class notes after completion of this lesson, after completing the class. Okay,
You
so
can
let's
go very
start.
slow, Anisha. The subject's contents, you can go very slow because we are foundation class students. So we have plenty of time in 10th standard. So go one by one, concept wise concept, and need not be in a hurry. Okay?
Okay, sir.
Okay.
Okay, sir.
Thank you.
Okay. So you must have read about the microbes lesson in class 9, right? Do you remember that?
Yes, ma'am.
Anyone?
Yes, ma'am.
Yes. So it's such an interesting lesson, you know. In NEET exam every year, at least 2 to 3 questions are asked from this lesson only. So that's why I have chosen this lesson. So today we are going to discuss some important topics which are related to NEET exam, particularly from the microbes in human welfare lesson of class 12 CBSE with some foundational information so that you can learn it easily. Okay. Besides macroscopic plants, we can see everything, right? Animals, plants, micro, everything. All are the major components of our biological system. So, do you remember who classified the biology based on the morphology? Do you know anyone know who first proposed the basis of simple morphological characters? Anyone know? Aristotle have proposed this classification on the basis of the presence of red blood cell and absence of red blood cell. In this basis, initially the scientists were unable to find such small organisms like bacteria, virus, fungi, protozoa, and many more things. You know, these are microbes are present everywhere. They are present in soil, they are present in water, they are present in air, inside our bodies, you know, on the body of the animals and on plants, and they are present even at the sites where no other life form could exist, like very cold areas, extremely cold areas. Even in our freezer there might be freezer, I mean, I am telling about the refrigerator, there might be some microbes living. And also microbes are very diverse, right? So even under the layers of snow several meter thick and in highly acidic environments, thermophiles do exist. So microbes like bacteria and many fungi, they can be grown on nutritive media, right? So these nutritive media, they are supposed to form colonies that can be seen with the naked eyes. But such cultures are not used that much nowadays, and people who are doing research in biotechnology and who are doing research in about fungi, about the microscopic plants like that, they just use those type of things. And those organisms which are not visible with naked eyes and can be viewed under the microscope are microorganisms or microbes. They are the major component of biological systems, right? So microbes like bacteria and many fungi can be grown in our daily life with the help of some simple experiments, right? So in this lesson basically focuses use uh, about the uses of microbes in household products and in and their usages like in antibiotics, in sewage treatment, and they are also used as chemicals, enzymes, and other bioactive molecules. And microbes are also used for the production of biogas. And based upon this, we are using methane. Methane microbes can produce methane, and microbes are used as biocontrol agents. They can be used as biofertilizers. and we can use them in wide spectrum and narrow spectrum applications. So first we are going to see about the microbes which are used in the household products. Do you know which microbe is present in the curd? Anyone?
Lactobacillus, ma'am.
Very good. Yes, that's the correct answer. Yes, lactobacillus. This is a very famous microorganism, right? We are commonly call it as lactic acid bacteria. It grow in milk and it converts it into curd. During the growth, the lactic acid bacteria produce acids that coagulate and partially digest the milk proteins. A small amount of curd, just a very small amount of curd is enough. If it is added to the fresh milk as an inoculum means it's a starter. Okay, so it contains millions of lactobacillic bacteria, and which can multiply at suitable temperature. So and it can convert milk into curd. What is the difference between milk and curd? Can anyone tell me? What is the difference between milk and curd? Anyone?
I know the difference.
Okay, sir.
Coffee
You
can be made with milk, but coffee cannot be made with curd.
I mean, chemically. I'm asking chemically, like.
Ma'am,
So
milk is a liquid, ma'am, whereas curd is a semi-solid, ma'am.
okay, yes. Physically,
A
you are saying
fermented product.
yes. You all are right. Physically, I'm asking chemically. So the chemically, the nutritional quality will be increased. That's the thing. So like in normal milk, there will be not that much vitamin B12. But in curd, it is like it will increase like more than 10 to 20 times. Curd is it's like we can make it into yogurt, and it has the good bacteria in our intestine, and it maintains the gastrointestinal tract of our body and many things. We people say that microorganisms are not good for health. I mean, like most of the microorganisms are heterotrophic. They just depend on other animals or plants or other living organisms to survive. But do microbes do exist which are autotrophic? They can even do photosynthesis, right? We know that cyanobacteria, yes, it is a type of eubacteria, true bacteria, which can do photosynthesis on its own, and it can survive, right? In main point of view, microbes names are very important. Most often the questions are asked from this chapter, they are microbe name. They will ask microbe name, what is which microbe is producing this, which microbe is producing that, like that. Okay, so basically lactic acid bacteria, it grow in milk and it convert into curd, and its nutritional quality is also increased. And also it play a very beneficial role in checking these causing microbes. Okay, can you tell which metabolic pathway is taking place, resulting in the formation of CO2 during the dough formation of making dosa and idli? Anyone? Can anyone say fermentation is happening, right? So if you in the dough, similarly the dough which is used for making bread, it is fermented by using the baker's yeast, Saccharomyces cerevisiae. Saccharomyces cerevisiae. Okay, so a number of traditional drinks and foods are also made by fermentation by the microbes. Okay, toddy is a traditional drink of some parts of southern India, which is made by fermenting sap from the palm trees. Okay, they will just ferment the sap and they will make the traditional drink toddy. Okay, and microbes are also used to ferment fish, soya bean, bamboo shoots, to make the foods, right? And cheese is one of the oldest food items in which microbes were used. Different varieties of cheese are there, different texture, different flavor, different taste, and the specificity coming from that is based on the specificity coming from the microbes used. For example, this is a very important point on the basis on the need basis. There is a cheese called a Swiss cheese, and in that Swiss cheese there will be very large holes when you compare to the like in Tom and Jerry we can see Jerry usually carry a piece of cheese which have very large hole, right? So that's nothing but the Swiss cheese. That how the cheese was produced is very interesting. There was a bacterium named Propionibacterium shermani. Propionibacterium shermani. Large amount of CO2 is produced by that bacterium. Due to that, we can see that much big, large holes in the cheese. And another microbe, which is a fungi, basically, which produces the Roquefort cheese. Roquefort cheese. The fungi is producing that, and that is ripened by growing a specific fungi on that, which give a very particular flavor for those kind of cheeses. And microbes are used in industrial products also, right? So they are on the production of them in a large scale. It requires very large vessels called fermenters. Fermenters are large vessels. I have included the pictures in the PDF. So in that vessels, they are cultured and they are developed so that they can be used for the industrial uses. And also some beverages, right? Wine, beer, whiskey, brandy, rum, everything is produced with the help of the microbe, especially yeast. Okay, yeast is a unicellular fungi, we know that, right? So for that purpose, the same yeast, the brewer's yeast, Saccharomyces cerevisiae, is used. We can ferment malted cereals, fruit juices to produce the ethanol. Okay, so the another very important point is wine and beer are produced without distillation. But whereas everything except wine and beer, whiskey, brandy, and rum, all are produced by the distillation of the fermented broth. Okay. So this question was asked in NEET exam, I think so. I don't remember the year. Wine and beer are produced without distillation, whereas whiskey, brandy, and rum are produced by the distillation of the fermented broth. And also,
this distillation by distillation you mean filtering?
what's that?
What do you mean by distillation? Is it filtering?
No, sir. It's like yes, it is nearly like purifying and clearing. Yes,
Oh, okay.
clearing. So yeah, they will have some. For example, in case of sugar cane, we will have some molasses, right? While extracting the sugar, in that case, we will remove that molasses.
Okay, okay. And when I go to market, my wife asks me to buy salty cheese, and some cheese is not salty. What is this?
That's based on the composition, sir, chemical composition. Some cheeses are salty made. We can add salt to the cheese as well.
Okay. Oh, okay.
Sometimes, yes, we can salty cheese. They are that's based on the taste and flavor, right? We can add flavors to cheese not only with the help of microbes. We can add artificially like that.
Okay. Then the doubt I have is what is the difference between yogurt and curd?
Yogurt and curd are nearly the same, but there is a difference in flavor. I think so. I haven't consumed it
Because
before,
in
but
US they don't call it as curd; they call yogurt.
Oh.
Sir, yogurt
here
is
in
it is
India we
somewhat
call it as
sweeter than
curd.
curd, sir.
Yeah.
we can add flavors, right? So we can add flavors. Yogurts are available in many flavors. They are basically prepared from the curry texture of the milk.
Okay. Thank you.
So yes, these fermented beverages are playing so these microbes are playing a very important role for making these kind of beverages. Okay, so this is in class 12 NCERT syllabus, and this is a very important NEET point of view topic. And then antibiotics. Antibiotics are produced by microbes. We know if anti means against, bio means life. So antibiotic means against life. Specifically, antibiotics will work for bacteria only, not for virus. So if we are having a throat infection or something like bacterial infection, we used to take amoxicillin tablet, right? Amoxicillin tablet. Usually, all people use that for as an antibiotic, but it won't work for fever or flu caused by the viruses. And we usually use that for throat pain, common cold, like that. So cold is not caused by virus, not caused by bacteria, but still we use that for the throat infection. Throat infection is usually caused by the bacteria. So we use the amoxicillin and that kind of medicines for that. So antibiotics are, they can be wide spectrum or narrow spectrum. Some antibiotics are specifically aimed to kill the particular type of bacteria. Some are usually used for to kill all type of bacteria, even it is a good or bad bacteria, they don't matter, they will kill everything. Okay, so antibiotics are chemical substances, okay, which are also produced by microbes, right? They can kill or retard the growth of other. You might be knowing about penicillin. Do you know who discovered penicillin? Anyone? Can anyone tell me who discovered penicillin? That's the first antibiotic to be discovered. Yes, very
Alexander Fleming.
yes, that's very correct.
Yes.
Okay. Kavinaya is raising a hand. Why?
No, ma'am, I have mistakenly touched it, ma'am, for instead of taking unmute.
No, I know why
Okay,
she
okay.
raised the hand. Our sister கிள்ளிட்டாங்க அதனால.
Oh,
No,
okay. So, so in case of Alexander Fleming, while working on a bacteria, this is a very important concept, you know. Staphylococci. You know, bacteria is having different shape. Coccus, we on the basis of shape, we can differentiate them. Coccus, Vibrio, and then rod shaped as Bacillus, and I have included that in the PDF also. So based on that, he was working on a bacteria, Staphylococci. Once he observed a mold growing in one of his unwashed culture plate. Okay. So unwashed culture plate means it is still having bacteria. Okay. Uh, where Staphylococci is, he found that it was due to a chemical produced by the mold. Suddenly, the Staphylococci cannot grow. He was very wondered. He wondered very like how this can grow here, the mole, how this can grow here like that. And he did research on that. However, its full potential as an effective antibiotic was established later. He found that something is present in that. The bacteria cannot grow. Why that bacteria is not growing on that culture plate? I use that culture plate for growing the bacteria. And later he observed that he had his Penicillium notatum mold in that. And then Ernst Chain and Howard Florey, two scientists, Ernst Chain and Howard Florey, they found that that is an effective antibiotic that can be used for people to treat many diseases, and that antibiotic was extensively used to treat American soldiers wounded in the World War II. And Fleming, Chain, Florey, all of them are awarded the Nobel Prize in 1945 for this discovery. After penicillin, many antibiotics were also prepared from other microbes. We can prepare antibiotic from one microbe to kill another microbe. They have greatly improved our capacity to treat deadly diseases like plague, whooping cough, diphtheria, leprosy, which is used to kill millions of people around the world. Today, we cannot imagine a world without microbes. Okay, microbes are small, but we cannot live without microbes. Even our bacteria, there are some good bacteria. They do exist. Without good bacteria, we cannot do digestion. In our intestine, there are more good bacteria and more bad bacteria as well. So the thing is, the key concept is when the balance of bacteria is not correct in our body, when the bacteria balance is not correct in our body, we will fall ill. The thing. That's why people usually say have more curd because curd usually multiplies the good bacteria in our body. Especially it keeps our gastrointestinal tract very healthy. Right. So we are using these kind of antibiotics to treat deadly diseases. Right. So also other than bacteria, we have virus, viroids, prions, and then lichens, and these are also microbes, right? So the name virus, that means venom or poisonous fluid, was first given by Dmitry Ivanovsky. Dmitry Ivanovsky was a scientist who recognized certain microbes as casual organisms of the mosaic disease of tobacco. These were found smaller than bacteria because they pass through the bacteria-proof filters. Later, a scientist called M.W. Bejernik demonstrated that the extract of the infected plant of tobacco could cause infection in healthy plants. And he called this fluid as contagium vivum fluidum. That means infectious living fluid. And again, a scientist came. His name was W.M. Stanley. He showed that viruses could be crystallized. Okay. So, and crystals consist largely of proteins. They are inert outside their specific host cell. Viruses are obligate parasites. In addition to proteins, viruses also contain genetic material that can be either RNA or DNA. No virus contain both RNA and DNA, right? So a virus that infect plants usually have a single-stranded RNA. Antiviruses that infect animals have either single or double-stranded RNA. Okay. So or even they can have double-stranded DNA. Okay. In general, viruses that infect plants have single-stranded RNA, and viruses that infect animals have either single or double-stranded RNA or double-stranded DNA. Usually, double-stranded DNA viruses they are mostly affecting the human beings, right? And the protein is made up of a small subunit called capsomere. There is a protein on virus that is called as capsid. It is made of small subunits called capsomeres. What is the function of this capsomere? It protects the nucleic acid. These capsomeres are arranged in helical, polyhedral, and many geometric forms to protect the subunit, to protect the nucleic acid. Viruses cause diseases, many diseases like mumps, smallpox, herpes, and influenza, and most importantly, AIDS. AIDS in humans is also caused by a virus. In plants, it can cause mosaic formation, leaf rolling and curling, yellow and vein clearing, dwarfing, and stunted growth like that. These viruses are in the don't have any use for human beings, but still people are saying they are used in genetic engineering. Even though virus is like a venom, it's like a poisonous fluid. People are saying it's like a venom. It is inert outside. When it comes into a host, it will take over the entire machinery of the cell. It will take over the entire machinery. It acts like a parasite. It is also used in genetic engineering. That is retrovirus. Retrovirus is used in genetic engineering. Okay. So if you want to modify a gene, you have to insert a vector, right? So this retrovirus, which is a very, very harmful virus. Okay. So they are used to inject a gene with a specific character or trait in the genetic engineering. And this is beyond the lesson. I'm just saying this. So, and also there is a particle which is smaller than virus. I'm saying this is a particle because they are not living, right? Viruses are not living. They are not even included in the plant kingdom, animal kingdom, anything. They are not even included in the 5 kingdom classification, right? So in 1971, a scientist named T.O. Diner, he discovered a new infectious agent that was very smaller than virus. That is smaller than virus, right? So, and it caused potato spindle tuber disease in potatoes, and it was found to be of a free RNA. It didn't have the protein code that is found in the virus. So he named it as viroid. The RNA of the viroid was of low molecular weight, so it didn't have that much weight. And in case of prions, in modern medicine, certain infectious neurological diseases are found to be transmitted by the agents, right? So it contained abnormally folded protein, and they called it as prions. This is a very, very serious condition, because if you are getting prions in our body, it is really very difficult to survive from that. Usually, it is very rare because protein. What will happen if a protein is infecting our body's mechanism? The most notable diseases caused are bovine spongiform encephalopathy. So we call it as mad cow disease in cattle. Once these kind of diseases they cannot survive easily. And the short form of it is BSE, bovine spongiform encephalopathy. And its analogous variant in human is called CR Jacob disease, CJD, CR Jacob disease. And also we have lichens. Lichens are symbiotic associations. They are mutually helpful. They are mutually helpful. They are mutually useful for each other between the algae and fungi. The algal component is known as phycobiont, and the fungal component is known as mycobiont. Mycobiont is heterotrophic. We know that, right? Fungi are heterotrophic. They depend on other organisms for living. Whereas algae, they are autotrophic. They can do photosynthesis. They can survive on their own. So both are helping each other. How? Algae prepare food for fungi, and fungi provide shelter and absorb mineral nutrients and water for its partner. So close is their association, like we cannot even imagine that there have been two different organisms within them. They look like that's like a one organism. We will think there is an organism, it may look like fungi, but actually there are two living organisms which are in mutual association with each other. So lichens are very good pollution indicators. They do not grow in polluted areas. Even if they are growing somewhere, if that area is polluted, they will change their color into red or mild orange, like that. We can easily say if you can see lichen somewhere, you might probably see in regions like mountains, pure and the places having pure air, humid air, like that. Okay. So, and these are the some overview about some microbes which are useful for human beings, for useful for making bioactive molecules, enzymes, and more. Another thing, microbes are also used for making the enzymes and bioactive molecules. They are used for commercial and industrial production of certain chemicals like organic acids, alcohols, and enzymes. For example, Aspergillus niger. Okay, so it is a fungus. It is useful for producing citric acid. Okay, Acetobacter aceti. It is a bacterium. Many of you know it is made. It is useful for making acetic acid. And then Clostridium butylicum. It is a bacterium. It is useful for producing butyric acid. And then Lactobacillus. It is a bacterium useful for producing lactic acid.
Lactic acid.
Yes. Microbes are also used for production of enzymes. Okay, lipases. What is a lipase? Lipid. What is lipid? It is a component of fatty acid, right? I mean, like it is a big molecular structure of fatty acid. Lipase means it is going to break. Okay, AS means break. Lipase, protease. We have more kind of A. If something is ending with ASE, then it is nothing but enzyme in case of biology. Okay, lipase, protease, nuclease, cellulase, like that. Okay, so lipases are used in detergent formulation and are helpful in removing oily stains from the laundry. You must have noticed in the bottle, notice that the bottle fruit juices are clear. Okay, they are much clearer as compared to those made at home, right? So if you are making juices at home, they are not that much clear. But bottle juices are like they are thick and they are clear. They don't have the molasses like things like in sugarcane, right? So this is because these juices are clarified by the use of pectinase and protease. Okay. And then another important, very important question. This is asked in NEET multiple times. This is asked in AIPMT exam, JIPMER, AIMS. This is a very important point. Please note this. Streptokinase. This is produced by the bacterium Streptococcus and it is modified by the genetic engineering and it is used as a clot buster. Blood clot occurs in the near the heart, right? So usually it occurs near the blood vessels which are near the heart. Okay, so this microbe is used for removing the clot from the blood vessels of patients who have undergone myocardial infarction. Okay, myocardial infarction leads to heart attack. So this is a life-saving bacteria, right? Streptokinase produced by Streptococcus. It is modified by genetic engineering. Another bioactive molecule called cyclosporin A. Cyclosporin A. This is a bioactive molecule that is used as an immunosuppressive agent in organ transplant patients, right? So if we are grafting an organ like kidney or people do grafting, so they usually prefer a close relative and they usually do some tissue matching test, blood matching test before organ transplant. But still, your body is very clever, right? Our human body is very clever. It can easily identify the foreign and its own organs, right? We have antibodies in our blood. It will easily identify the that this is not our kidney, this is not our eye, like that it will think, and it will send the antibodies to kill that blood flow, to kill that particular organ, right? So to prevent this, our body is infused with immunosuppressant. So these immunosuppressive agents, so they will suppress the immune system, right? So that is cyclosporin A. Cyclosporin A is extracted from the fungus Trichoderma polysporum. Okay, this is a very important aspect in NEET aspect. And statins are produced by the yeast called Monascus purpureus. Okay, this has been commercialized as blood cholesterol lowering agent. It acts by competitively inhibiting the enzyme responsible for the synthesis of cholesterol. People are developing cholesterol due to poor health, poor diet, lack of exercise, and maybe cholesterol accumulation can also be even linked with the genetics, right? Genetically can also, some people might say this. Genetically, they have a fat body like that. Okay, so in that cases, we cannot do anything, right? So we have to use these kind of drugs to prevent competitively inhibit the enzyme. What do you know? Anyone know what is competitive inhibition? Do anyone know what is competitive inhibition? Okay, let me explain. Competitive inhibition means usually for synthesizing a cholesterol, there is an enzyme. So, for that enzyme, for example, that enzyme is having a structure like a key. Okay, key like structure or a cloud like structure. Just imagine. So instead of enzymes usually have some sites where those kind of those kind of receptors do join, and we will send the competitive inhibitor. Competitive inhibitor will also have the same structure like the enzyme. In that case, the body cannot recognize which is the enzyme, which is the competitive inhibitor, like that. So suddenly, if competitive inhibitor inhibits the joining of the enzyme in the particular receptive site, so the process is controlled and cholesterol is lowered. Okay, so this is the mechanism behind it. It is not that much needed, but I am just saying because we have to know what is competitive inhibition, like that. In NCRT, we are not that much. So in class 12 CBSE, they are just saying everything by like they just touch the periphery, not just like in the Samachit books of TN State Board. They just touch the periphery. So sometimes we end up confusing everything. Okay. So microbes are used in sewage treatment also. Okay. Large quantities of water, wastewater are generated every day in cities and towns. Major component are human excreta, and the municipal wastewater is called as sewage. We know this. So it contains large amount of organic matter. And also they have microbes which are harmful, many of which are pathogenic. Wastewater should be disposed of in a very clean manner, right? So we have to process them. We have to process them. So they cannot be discharged into natural water bodies like rivers and streams directly. You know why? Before disposal, hence sewage is treated in sewage treatment plants. We call them as STPs to make it less polluting. Treatment of wastewater is done by the heterotrophic microbes naturally present in the sewage. Okay, this treatment is carried out in two stages: primary treatment and secondary treatment. I have given a mind map which you can easily remember everything in that PDF. That mind map will include the steps of treatment, primary treatment, and secondary treatment. Primary treatment is also called as mechanical treatment. Secondary treatment is called as biological treatment. There are chemical treatments too, but they are not included in our syllabus. Chemically, even after biological treatment, some like we are chlorinating, right? So we are chlorinating water. That's like the that's out of syllabus, but there is also a chemical treatment which exists as the third. From the sewage to filtration and sedimentation. So initially we will it's like picking up the pebbles, soil. They are usually removed by sedimentation. Okay, all solids that settle form the primary sludge. Okay, and the supernatant form.
Anisha, I just have one doubt. What is this BOD and COD? Can you do you know BOD and COD?
Sir, BOD means biological oxygen demand, sir.
And
It refers to the.
yeah, can you
I don't
explain to
know about.
our children? Can you explain to our children in a sewage or in a water? They always talk about BOD.
Yes, sir.
Uh, uh,
BOD is a BOD means biochemical oxygen demand.
hmm,
Okay. So BOD refers to the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by bacteria.
hmm. Okay. So have you
In a sample,
children? Have you heard of this BOD, BOD, COD? Because in
No, sir.
any potable water, drinking water, they always call BOD is this much and things like that. I don't know exactly. I'm just asking a layman's question. Maybe go through your books. Maybe next class Anisha can deal with it in detail. Okay, Anisha. I mean, this may
yes,
not be a neat question.
yes, sir.
This is a general, no, a general thing which we read in our newspapers and things like that. No, from that I'm asking. Okay, carry on, carry on.
Yes, sir.
Maybe you can
So first, for
carry on, carry on, carry on.
okay, sir. So if you are taking this digester that will have the anaerobic, both anaerobic and aerobic stuff. Sir, wait, sir. I will try to screen share. Now it's showing me. Sir, is my voice visible?
Yeah, your voice is audible. Your video is not visible. Video came and gone. Only Junior Anisha is visible.
Okay, sir. I already told you I have some problem with the screen sharing. Maybe in
Yeah,
future I
no,
will
no,
buy a laptop.
next class, no, we will manage. No problem. Next class, I will also, send small files. Then I will try to, share the screen for you.
Okay, sir. Okay,
Okay,
sir.
okay, okay. Right. This class also.
So here,
Yeah.
in the case of primary treatment, it's a physical process of removal of small and large particles through filtration and sedimentation, right? So by letting the sewage to pass through via mesh screens of smaller pore sizes, floating substances like leaves, polythene are removed sequentially. Then the sewage is passed on to a grit chamber where the grit means soil and pebbles. They are sedimented there, and the sewage is allowed to go into a primary settling tank. The suspended material settle down to form the primary sludge in the settling tank. Okay, so flocks means they are masses of bacteria associated with fungal hyphae that form mesh-like structure. And biological oxygen demand, I have already told you that it indicates the amount of organic matter present in the sewage water. And in case of secondary treatment, I have told it already that it is a biological process. It is carried out by the bacteria present in the water, present in the sewage itself, right? So, in case of biological treatment, first the effluent from the primary treatment is passed into large aeration tanks. Here it is constantly agitated and air is pumped into it. And the agitation allows rapid growth of aerobic microbes into flocks. I have just told they are masses of bacteria associated with fungal hypha. Okay, that is what we call as flock. Okay, so these flock digest the organic matter of the sewage and they reduce the biological oxygen demand. Once the BOD of the sewage is reduced significantly, the effluent is passed into a settling tank where the flocks are allowed to sediment forming the activated sludge. Then a small part of activated sludge is pumped back into the aeration tank as inoculum. Then the remaining part of the sludge is pumped into anaerobic sludge digester where the anaerobic bacteria digest the bacteria and fungi in the sludge producing biogas. Do you know what are the gases present in biogas? Anyone? Can anyone tell me? Any one gas?
Methane?
Yes, methane and two gases left. Can anyone?
Carbon dioxide, ma'am.
Super. And another gas is left. Can anyone tell that too?
Oxygen?
Ma'am.
No.
And then
How there would be
smaller amounts of hydrogen sulfide and ammonia, ma'am.
very good. Yes. This is called very good, very good. Yes. So yes, we are having methane, hydrogen sulfide, small amount of ammonia, and carbon dioxide. Thank you. Who told that?
Myself, Kavinaya, ma'am.
Very good.
No,
Thank
initially
you.
somebody else told, I think. Initially somebody else. I don't know who is that.
So
Anyway, no problem. Yeah, carry on, you know.
yes, and
And
also
I have a request to Anisha. So you have another 10 minutes. Last 5 minutes, you handle the class in American English.
okay, sir. Sure, sir.
Okay.
So I have really slowed my speaking more. That's because people will not understand. That's why. Okay. So let's see. Let's go further. So the fungus trichoderma is a free living in the soil and root ecosystems, and is used against several plant pathogens, right? So baculoviruses are pathogens that attack many insects and arthropods. One of the baculovirus used as a biocontrol agent is nucleopolyhedrovirus. They are species specific and do not harm other pests, plants, or animals. So if you are taking microbes as fertilizer, they are biofertilizers or microorganisms that are used to enrich the fertility of the soil. They increase the fertility of the soil by adding certain nutrients to the soil. Three groups of organisms are used as biofertilizers. They are bacteria, cyanobacteria, and fungi. So fungi, usually they are mycorrhiza. So bacteria, they are the following types. First thing is symbiotic bacterium and loose association of nitrogen fixing bacterium and free living bacterium. Symbiotic bacterium, if you are telling about symbiotic bacterium, that is rhizobium, right? So rhizobium shows a symbiotic association with the root nodules of leguminous plants. It fixes nitrogen into soil and it increases its fertility. It forms nodules in the roots of plants, and also they are used to show symbiotic association with other fungi as well. With soybean, Rhizobium japonicum, and with clover group, R. trifoli, with alpha alpha, R. meltiloti, with bean group, R. fascioli. People don't feel bad because I have entered these details in the PDF, because while speaking, these names might look awful, right? So, loose association of nitrogen fixing bacterium, in that case, certain bacterium live around the roots of higher plants, but they do not develop any relationship with the plant. The bacteria obtain some exudates from the plant and they use it as the food. For example, we can take Azoospermillum. In case of free living bacterium, they live freely in the soil and fix nitrogen. And the examples are Acetobacter, Bacillus palmixia, and Beigernica. Cyanobacteria. One, symbiotic nitrogen fixing bacteria, and the two is free living nitrogen fixing cyanobacteria. If you are taking symbiotic nitrogen fixing bacteria, they form symbiotic association with certain plants. Azolla anabina association is an association of the leaves of the fern Azolla pinnata with cyanobacteria. Anabina lives in the corallite roots of cycas. Azolla and nostoc are added in the rice fields to increase the rice yield.
Maybe
And free living.
you can wind up in another 2 minutes. And before you
Okay.
wind up, you can ask one or two questions on whatever you taught, and then you can wind up.
Okay, sure, sir. Free living nitrogen fixing bacteria is left. Some cyanobacteria live in the soil and fix nitrogen. They add organic matter and also nitrogen to the soil. Example, Aloe fertilissima is a nitrogen fixer in the rice fields. If you are talking about mycorrhiza, they are fungi form symbiotic association with roots of higher plants. Example, glomus. They are of two types, ectomycorrhiza and endomycorrhiza. Okay, so this is just an overview of this lesson. There are many topics which can be covered. I have told about those things which are basic for NEET examination. This is just a foundation. We can do research for many. For example, even if you are looking into the Samachir book, TN Kalvi Samachir book, we will find many interesting thing about virus and the airborne diseases, waterborne diseases like that. So let me ask you a few questions. Are you ready? Everyone, tell me. Are you ready? So, okay. Uh,
Yes, we are ready for the question.
can anyone? Okay. Which type of what are the beverages which are prepared without distillation?
I know the answer.
And all the students have to try. Which type of beverages are prepared without distillation?
Ma'am, could you please repeat the question again?
Which type of beverages are prepared without distillation? Okay, I will give you option.
I will give
You
you
can try. Option one. Okay, sir. You give the answer, sir.
beer and wine.
Wow, yes. This is a very important question in the aspect.
No, this is, students, I just interfered
Okay.
in your class for simple reason. That is sometimes listening, if you have a focus, you can listen and, and grasp whatever is taught. I was actually coming and going, but then this particular part I was listening to this. Immediately after this, I asked one question also: what is meant by distillation? And my understanding is only filtering. So like that. So that is the reason I remembered. So you can listen even without seeing the person. Also, you can listen. A little bit of more focus is required for you to grasp what is taught. Okay, thank you. Yeah, carry on.
Okay. How many steps are in CH treatment? Can anyone tell it?
Can you repeat the question?
How many steps are in the sewage treatment? Anyone? Types? Can you narrate the types of treatment?
Ma'am, there are three main stages, ma'am.
Okay. What are they?
Primary, secondary, and tertiary treatment, ma'am.
Super, excellent. In NCRT, we only have primary and secondary treatment. So if they ask in NEET exam, write two. But actually, there are three treatments. Write as true, write as two in NEET exam. Okay.
Yeah, yes.
Okay. What is the another name for secondary treatment?
Is it aeration?
Anyone?
Aeration?
No. It is based on the type of treatment.
Oh,
Anyone?
Ma'am, biological treatment, ma'am.
Wow, super. Yes. Okay. So, what are the gases which are produced in the biogas production? Anyone?
Ma'am, methane, carbon dioxide, and a little amount of ammonia and sulfur dioxide.
No,
hydrogen
not
sulfide.
sulfur dioxide.
Hydrogen
Hydrogen
sulfide.
sulfide.
Sorry, hydrogen sulfide.
No, this hydrogen
Very
sulfide smell will be equivalent to rotten egg smell, no? Am I correct?
Yes, sir. Yes, sir. Yes,
Yes,
sir. You are
ma'am.
right.
Yes, sir.
Oh, very good. My memory is better.
Yes, yes,
I
yes.
learned it 50 years back.
Wow. Yes, sir. Okay. Uh, then what can I ask?
Now, I will ask students to ask questions. Students, you can ask 1 or 2 questions to madam, to Anisha. But Anisha, I have one question. You have talked about fermentation. After
Yes,
fermentation,
sir.
the item will become a coagulation or something.
Sir.
Huh?
No, it will not coagulate, but it will look like coagulated. Actually, in fermentation, there will be a release of carbon dioxide, right? So there, the gap will be produced. Okay, there will be some kind of gap produced. We can easily say by just looking whether it is fermented or not. Just imagine the dosa or idli batter.
Mm.
Okay, before fermentation it will look thick, after fermentation it will just foam up, it will
Correct.
foam up, it will come out of vessel.
Correct.
That's what I told, sir.
Okay. Okay. Right. Anyway, thank you, Anisha, for your class. Two suggestions I have for you. Of course, positive things, we know each other, so I need not talk about positive things about Anisha. I mean, she is very good in subject, very good with the language. These are the positive things. Second thing is you can go a little slow. No, there are,
Okay, sir.
because these students are 9th and 10th standard students. Obviously, you need to go a little slow, and that also will help you your breathing also. No? When you go slow, you can breathe properly, and then do that. And then and there you can ask questions. Then and there, the students, you can make students to ask questions. That will help you in this thing. And the last 5 minutes she was talking, she is trying to talk in American English. Are you able to see the difference, students?
Yes, sir.
Yeah, so she's good in it. And I would suggest her every class, last 5 minutes, you speak in such a language, so that our students will be enthused in your class to listen to you, as well as your English language command. So, any questions if we have? Otherwise, we can wind up. Sachin Patil, what is your comment? Palak, I mean, her name is Palak. Palak, are you there? Oh, she's not there. Aditi,
Yes, sir.
you want to say something? You came late.
Best class, sir.
Yeah, you came late. Okay, no problem. Srinidhi, you want to say something?
No, sir.
Srinidhi, this teacher is from Tiruvarur, and Srinidhi is from Tiruthurai, much closer to Tiruvarur, about 30 kilometers. Okay, thank you, all of you. Keep coming. Anisha will continue to help us, support us in a big way. And Anisha was our student about 3-4 years back. Anisha, 3 years back.
Okay, sir. I am here from the beginning itself, sir.
Yeah. See, 4-5 years back, she was our student for continuously for two years or so. Then later on, no, she went into studies and other things. So Anisha is student. Anisha's younger sister was our student, and our younger brother also was our student. Just for information, the whole family was our student. Okay, thank you,
Hmm.
Anisha. I will keep in
Okay.
touch, and next time we will share the screen. Okay,
Okay, sir. Thank
okay,
you, sir.
without any problem. Yeah.
Thank you, sir.